Okay, thank you, Jessica, for your introduction and for all the organizers for the organization of this OCB webinar. I'm Zhang from Shenzhen University. Actually, I just moved from Xiamen University last year. Basically, my research focuses on marine water ecology. So today, I would like to share some stories about the ecological and biochemical aspect of marine viruses, particularly focus on their life strategy. Okay, viruses are a group of ultra-microscopic infectious biological particles. If we put viruses in the context of marine life forms, as you can see here, they are the smallest one on the edge of all life forms. The average size of marine viruses is about maybe 100 nanometers, and the smallest one maybe only less than 20 nanometers. So there are about 10 orders of magnitude difference in size from viruses and from the big mammals like blue whale. So they are very small. But we always said small is beautiful. So this is a picture showing typical marine phages viruses infecting bacteria. We use the cryo-EM to take photos for it. And you can see a beautiful symmetric pattern, right? So most of the phages have a protein capsid containing a DNA or RNA genome and a tail that is used to attach to the host cell surface. So compared to cellular life, viruses are very simple, but simple is the best. So someone says viruses are maybe the most successful inhabited on Earth because they are the most abundant biological forms. If we take a drop of seawater and filter it on a membrane, stain the membrane with DNA dye and observed under epifluorescence microscope, you will see this image. And these big dot cells, basically they are maybe from bacteria or archaea, or maybe some phytoplankton. And this little, right, this little dots, they are viral particles. So on average, there are about 5 to 10 million viral particles in every milliliter of seawater. And the total number of viruses in the ocean is about 10 to the power 30. So that's a lot. So the ecological role of viruses in the oceanic ecosystem depend on their life strategies, mainly include lytic and lysogenic cycles. And based on this, we can divide them into two groups: virulent phages and temperate phages. Virulent phages displayed only lytic cycle, so the only way to grow is to kill the host and release new viruses. Temperate phages, however, can enter either lytic or lysogenic cycle in which they integrate their DNA into host genome and become prophage. So under certain conditions or with some inducers, prophage can be induced and enter lytic cycle. So some people call it a time bomb in the host. We will discuss this later. So let's see lytic infection firstly. Virus actually they can infect and lysate all ecological components, including bacterial plankton, phytoplankton, and zooplankton. So playing a role of top-down control for them. And every second, there are about 10 to the power 23rd infection taking place in the global ocean. So this leads about 1 out of 3 cells being killed per day. So viruses play an important top-down control for microbes in the ocean. Meanwhile, the lysis process releases cellular content, including proteins, amino acid, and lipid. And these materials are quite labile, so they are readily reused by other bacteria. In this case, lysis can be considered as one of the bottom-up factors for heterotrophic bacterial plankton. So this brings us to the concept of wire shunt, which is important for oceanic material cycling. By lysing cells and converting their cellular materials into bioavailable DOM, viruses disrupt the flow of organic carbon and nutrient from microbursts to high trophic level. So this results in more organic carbon staying in the DOM pool and being respect, decreasing the efficiency of energy transfer through the marine food web. Meanwhile, during the successive recycling of DOM, recalcitrated organic carbon would accumulate and be stored in the ocean for a longer time. So since there are numerous studies investing in wire lysis, I would like to just show two examples from my lab. Firstly, we studied the optical characteristics and the microutilization of the viral lysate of picophytoplankton Procococus. We found that DOM released or produced by lysis with three different features had higher humic-like fluorescence intensity, lower absorption coefficient, and higher spectral slope than DOM directly released by Procococus. So while infection altered the composition of Procococus-derived DOM and might contribute to the humic-like DOM pool in the ocean. And incubation with viral DOM resulted in a greater DOC degradation rate and a higher heterotrophic bacterial growth rate. This suggests that viral DOM was more bioavailable. In addition, the stimulated microbial community succession was significantly changed by viral DOM, which indicates that viral lysate play an important role in shaping the heterotrophic bacterial diversity. Then we found that picocyanobacteria of viral DOM showed similar apparent fluorescence yield and undergoes similar photodegradation behavior compared with deep sea DOM. So this is quite interesting. And ultra resolution mass spectry and AMR analysis showed that abundant nitrogen-containing compound in viral DOM. So they may be the degradation products of the fluorescence phycobilin pigment. So therefore, our data indicated that viral lysis of picocyanobacteria are likely to be important source of marine DOM, and viral lysate may accumulate in the deep sea and contribute to long-term carbon sequestration in the ocean. So if we look at the role of viral lysis at the oceanic scale, about 1/5 of the primary production is returned to the ocean DOC pool through viral shunt. So as shown in this simple model analysis, if there is no virus in the ocean, the direction and the flux of marine carbon cycling will be totally different. And then if we zoom out and put in viruses in global carbon cycling, we know that every year the ocean absorbed about 93 gigaton carbon and released about 90 gigaton carbon. So the net carbon flux is about 3 gigaton. Surprisingly, viral lysis release 150 gigaton of carbon per year. So this is almost 50 times the annual net carbon flux of the ocean. So marine virus was called nanoscale drive for global scale process. And also please note that this 150 gigaton is calculated based on limited viral lysis data in euphotic zone only, since there is no enough data for deep sea 20 years ago. Then, in order to have a more complete view of viral lysis in marine carbon cycling, we developed a global viral oceanography database, GVOD, which includes more than 10,000 of viral abundance data and more than 800 of viral activity data from published papers. Okay, now let's look at what extent we know about viral lysis in the ocean. Firstly, from the surface to the deep ocean, the lytic activity of the virus decreased by almost an order of magnitude. And viral lytic activity is significantly positively correlated with bacterial activity and abundance. So this fits the so-called kill the winner theory. This theory said the ecosystem with a higher bacterial abundance or activity would support a higher level of lytic viral activity, since viruses prefer to lysate the more dominant and rapidly growing bacteria. So we will come back to this in a few minutes. So based on our database, we can calculate that at the whole oceanic scale, while lysate release more than 400 gigaton carbon per year. So although there is a large uncertainty due to data limitation, there is no doubt that viral lactic activity greatly impact the marine carbon cycling. Then now let's move to the other life infection strategy, lysogenic cycle. As I mentioned before, temporophages integrate their DNA into the genome of the host bacteria as prophage and replicate with the host without causing cell death until lactic cycle is induced. So lysogenic infection actually they can alter host phenotype and physiology by expressing the genes in the viral genome or by horizontal gene transfer through transduction. So compared to non-lysogenic cells, lysogens always confer more fitness in terms of, for example, competitiveness, substrate utilization, biofilm formation, or maybe the resistance to environmental stress. In addition, establishment of lysogeny always confer immunity against the lactic attack by the same or close viruses. So this will help the domination of the bacterial community by lysogeny. Here I show one example from my lab study on central microbiome. It's a bacteria widely distributed in the ocean. We identified many prophages from central microbial strains collected globally and found that they contribute to bacterial microdiversity by shaping the evolution, co-evolution of bacterial pangenome and cogenomes. So we performed a large-scale cross-infection experiment, and this showed that all lysogens are immune to their own induced phage or closed phage. So this is a phenomenon we call the super-infection immunity. And the prophages are released and kill or invade susceptible strains. As shown with this cartoon, they are like a biological weapon to kill bacterial competitors. So it's beneficial for the host. So most of the previous studies of the viral live infection actually focus on lactic infection. But very limited studies have been performed for lysogenic infection, particularly at population and ecosystem level. So for a long time, lysogen has been proposed to be a preferable survival strategy for both the virus and the host in unfriendly environment. So based on the kill the winner theory, a higher level of lysogenic viral production usually appeared with a lower bacterial abundance or activity. However, a newly proposed piggyback the winner model suggests that lysogeny would also be interestingly favored with a higher microbial density or activity. So more work is needed to understand the viral lysogen pattern in marine microbial ecosystem. And recently, a global metagenomic and viromic analysis demonstrated that the lysogen ratio increased with water depth, reaching more than 50% in deep sea. So therefore, this data based on in silico analysis, the dry experiment showing that lysogen is probably more prevalent in harsh environment such as oligotrophic open ocean and the deep sea, where there is a low host abundance and activity. So this fit the so-called kill the winner theory. Then we checked the measured data of lysogenic activity with wet experiment, the real measurement. Again, our global viral oceanographic database provide a horizontal and a vertical distribution of lysogenic viral activity on global scale. Generally, it shows similar trend with lactic viral activity. But interestingly, we found the estuarine and the surface ocean showed higher lysogenic viral activity and the fraction of lysogenic cells. So compared to open ocean and deep sea. So this is a kind of piggyback the winner pattern. So on global scale, whether the viral life strategy follow kill the winner or piggyback the winner, it is still an open question. So it is, but it is definitely a complex issue because it's related to many factors such as host abundance, host activity, host diversity, virus to host ratio, and the environmental conditions such as nutrient level. And to complicate the matters further, viral life strategy can be switched. As I mentioned before, temporal phage may choose lysogenic infection under certain conditions, while the change of condition may induce lysogenic infection into lytic cycle. So one example is the transplant experiment that we did in South China Sea to simulate environmental changes during particle sinking and investigate their effects on viral ecodynamics. We performed 18 transplant experiments at 6 stations at the coastal and open oceans area. So at each station, surface bacteria and viruses were mixed with virus-free seawater of deep layer. So we found increased viral production, but decreased virus-mediated bacterial mortality after transplant. And we found a larger burst size, and the switch from the lysogenic to lytic strategy was shown to be to contribute to the enhanced viral production. So life strategy of surface virus changed dramatically after they sink into deep sea water. And the other example is a microcosm experiment in an estuarine environment. We explored viral life strategy and its potential effect on bacterial community and lysogen with lysogen-induced and non-induced treatment. And firstly, the lysogen-induced treatment showed relatively consistent viral and bacterial abundance and consistently lytic and lysogenic viral production throughout the incubation period. Conversely, the non-induced treatment displayed a marked variation in the abundance of viruses, total bacteria and active bacteria abundance. So this variation was congruent with the shift in lytic and lysogenic viral production. Sorry. And the viral, as well as the succession of bacterial community. So the lysogen to lysis switch may provide a growth opportunity for less active or initially rare bacterial species and generate a more diverse bacterial community. And we then did a time series observation for 3 stations with contrasting nutrient level in this estuary area, which covered a complete spring nip tide cycle. I may not have the time to go to the details, but we found that the distinct tidal dynamic pattern of microbes were tightly associated with the variation of viral production and decay. For example, in nip tide, we observed that low viral production, higher viral decay, lower bacterial abundance and activity. So this pattern was coupled with a switch from virolytic to lysogenic infection induced by tide mixing. So we have this model in estuarine environment. When the river regime dominated the estuarine ecosystem during nip tide, the low activity of bacteria together with a high level of viral decay rate jointly lead to the viral infection inefficiency. So in the lysis to lysogen switching, and this result finally resulted in a lower viral production. But during the springtide, the higher bacterial activity and low viral decay resulted in the lysogen to lysis switching, and finally higher viral production. So we did a similar time series monitoring at the coastal and the ocean stations on diel scale in South China Sea. And the diel variation in bacteria activity, bacterial community composition and diversity was also found highly affected by viral dynamics. The bacterial diversity was positively correlated to lactic viral production in the euphotic zone of the open ocean, but was negatively related to lysogenic viral production in the coastal area. So such distinct correlation suggests that viral life strategy can not only contribute to bacterial diversity, but also potentially piggyback their host to dominate the bacteria community. So I used this figure to show our major findings from this time series data set. It's a large data set. So in the coastal ocean, low viral lysis and high viral decay is observed during the daytime, while the opposite pattern appeared at nighttime. So high nitrogen is linked to the bacterial diversity in the coastal ocean following the scenario piggyback the window, and suggests that a viral nitrogenic lifestyle can potentially help the host dominate the bacterial community. In the open ocean, the diel pattern of viral lysis and decay at the surface water is similar to that of coastal water, and the virus host interaction at the subsurface water showed a different scenario. Higher viral lysis is linked to higher bacterial diversity, indicating that viral lysis can diversify the bacterial community as suggested by piggyback the window, kill the window model. So therefore, in nature, the proportion of virus in lactic and lysogenic infection vary across different environment conditions and different ecosystem or different time and space. But no matter to kill or not to kill, or no matter kill the winter or piggyback the winter, virus strategy play an important role in shaping microbial ecology and biogeochemistry. So maybe let me end my talk with this figure again. As I mentioned before, viral lactic infection may release more than 400 gigaton carbon per year. So then we calculate how much carbon held by lysogen. It's about 200 gigaton per year. So both are big numbers compared to the net annual carbon flux between air and sea. So it is clear that both of the life strategy of virus strongly impact our global carbon cycle. And I will end here. I will stop here. Finally, I would like to thank my lab members, collaborators, and funding agency for their support. Thank you.
Thank you. We have like a few minutes for question if you have like burning questions, and we'll have another opportunity at the end of the session. I do not, you can raise your hand or you can write in the chat. I don't, oh, I see one hand raised. Elena.
Oh, hi there. I had to jump
Hi.
in late because of a work meeting, but from what I caught, part of, oh, can you hear me? Sorry.
Yes.
Yes.
Okay,
Yes.
one of my headphones. I was curious about the switch from lysogeny to lytic and what's more important. And I saw a paper looking at lifestyle strategies. It was from several years ago, but it was showing that like microbial mortality was more likely from grazers, or it was like kind of shared between viruses and grazers in the surface, but then mortality was more viral as you go deeper. And I was just curious how that aligned with lysogenic versus lytic death. And yeah, if that was in line with what you saw as depth increased.
Yes. There are two major top-down control for marine microbials in the ocean. It's grazing and virulysis. And as you said, their relative contribution changed along maybe the nutrient gradient or the vertical gradient from the surface to the deep ocean. Because in the deep ocean or some low oxygen surface area, maybe the activity of the grazers is quite weak. So compared to the grazing, in this case, the virulysis is maybe a major factor for the mortality of the bacterial plankton. So But it's different. Actually, it's different scenario or how to say, it's different scenario for the lysogen and lytic viral activity. I think because gradient and lysis, the contribution of gradient and lysis, it's depend on for example, the room lectin. But for the lysis and the lytic and the lysogenic infection of viruses, it mainly depend on the host, on the prey, for example, like bacterial lectin. So I think that's the major difference between these two comparisons.
Okay, thank you.
Thank you.
Yeah.
And I'm going to pass the baton to Sherry, who will introduce our next speaker.
Hey, thanks, Jessica. And also thank you, Ray. That was a really great talk. I appreciated you highlighting a lot of the challenges we have in marine virology with quantification across global scales, and then also the challenge of understanding lysogeny and the switch between lysogeny and lytic and what's driving that and how that impacts global scale carbon cycles. It's not an easy thing. So I'd really appreciated that. So I'm very pleased to introduce Flora Vincent, who's our next speaker. Flora received her PhD in 2016, and then she did a postdoctoral research at the Weizmann Institute. And most recently, since 2022, she's been a group leader at EMBL, and her group focuses on understanding diversity and impact of marine microbial interactions across biological scales. She has a focus on symbioses within unicellular eukaryotes, but has done a lot of work understanding virus-host interactions. The work I'm most familiar with is in Emiliana Huxley, and she's going to talk with us about some of her new single-cell approaches today. So I'll hand it to you, Flora. Thank you.
Yes, thank you. You should be able to see. Can you see the presentation in full screen?
Yes, looks great.
Great, amazing. I'm just going to put a timer. Yeah, cool. So thanks a lot, Rui, for the first part of the talk, and also thanks to the OCB webinars for hosting me. And yeah, today I will try to share a bit some of the, I would say, latest work from the postdoc around single-cell ecology and how we've been using single-cell approaches to try and better quantify the impact of viral infection in the ocean. So yeah, most of what I will present is from the postdoc. So yeah, as you all probably know, every year one of the most, I think, outstanding phenomenon occurs in the ocean, whereby phytoplankton start to bloom. We know that they come in a wide diversity of shapes and sizes, but not only are they forming those massive blooms, they also play a major role for our ecosystem as primary producers. And back then, when I was, when we think about the fate of phytoplankton in the marine system, I would say that the classic food chain approach suggests that phytoplankton will be eaten by grazers, and this will transfer the carbon of algal biomass up the food chain. We know that phytoplankton can have different types of fates. For instance, when cells lyse, their cellular content can actually feed a pool of particulate and dissolved organic matter, which will trigger what we call the microbial loop. And in this loop, bacteria recycle algal biomass that will then be fed upon by a higher organism, and this kind of keeps the carbon in the surface part of the ocean. We know that phytoplankton can also produce sticky acidic polysaccharides leading to aggregation. Those aggregates form particles that can sink, in which case the carbon contained in algal biomass will be buried into the deep. However, this picture was kind of expanded a few decades ago with the discovery of viruses. Same picture than what Rui showed, a natural seawater stained with double-stranded DNA. All the small green dots are viruses that were quickly estimated to be the most abundant biological entity in the ocean. And I think this kind of really kickstarted the field of aquatic virology, and the emergence of new concepts. One of them we've heard of already quite a lot, which is the viral shunt. So the idea that a viral infection will enhance the lysis of algal cells, and this will actually lubricate the microbial loop and increase bacterial growth, with the idea that viruses basically shunt algal biomass away from eukaryotic grazing towards bacteria. But the viral shunt was also expanded to include what we now call the viral shuttle, which is the idea that upon viral infection, phytoplankton are going to aggregate even more, and that the biomass sinking is going to be increased or / the biosequestration of carbon into the deep. And I think that the big question, at least that I had when I started in this field of aquatic virology, was how much does viral infection really affect those two processes? Processes that probably occur quite often, given the abundance of viruses in the ocean on the one hand and the magnitude of algal bloom. So for me, it was really trying to better quantify that impact that I anticipated would be significant. So one of the main bottlenecks that we identified back then was the fact that it's quite hard to accurately measure the extent of viral infection in the natural environment. Traditionally, we would stain double-stranded DNA in natural samples, but the problem is that some of that stain is quite unspecific. It can stain vesicles, inactive particles, and it's not very specific. Later on, we started doing gene fish, so in situ hybridization targeting specific viral DNA. But the problem is that you don't really know if the virus is actually actively replicating versus being lysogen, for example. More recently, we started implementing bulk transcriptomics, which was actually quite useful because you can see which are the viruses that are active. But because we are basically squishing a complex community on a filter, it's hard to know who is the native host, and it's hard to know how many cells are actively being infected. I joined a SAF's lab at the moment where single-cell approaches were really starting to boom. And I hope that by the end of the talk, you will be convinced that it's a really nice tool, that single-cell approaches are a really nice tool to address some of those questions. So in a SAF's lab, we work on a host pathogen system that spans several orders of magnitude. And we exposed and studied that system across different scales of experimental setup in the very controlled laboratory setting, but also mesocosms, and finally reaching out to open ocean experiments. So the host is called Gephyrocapsa huxley, used to be Emiliana huxley, but now we say Gephyrocapsa huxley. It's a unicellular eukaryotic phytoplankton enveloped in this calcium carbonate cell wall. It can form those massive blooms every year that are visible from space. And these blooms were really thought to be terminated by a viral infection involving a giant double-stranded DNA virus called EHV. And this is really the main mortality agent, or considered as the main mortality agent. But the problem is that we couldn't really solve how many cells were actively infected during a bloom. And so starting from this very simple question, as long as we cannot answer that question, it will be very hard to evaluate the true impact of viruses on phytoplankton blooms. So to try and address this, we started actually in the lab using a very controlled setup of host and viruses co-culture, and decided to use in situ hybridization, labeling individual molecules of both viral and host messenger RNA, and to do this at the single cell level, and to acquire it not using microscopy, but using imaging flow cytometry. So this is exactly what you see here. I will try to put my pointer. This is what you see here, where every row is a cell. First, there's a bright field image, and then using a combination of excitation and emission laser, you can see whether or not your cell contains viral messenger RNA or host messenger RNA, or both. And because this is imaging flow cytometry, it's actually quite quantitative. Viral messenger RNA for us is important because the double-stranded DNA virus enters the cell and it starts to replicate. So when you get viral messenger RNA, it really means that your cell is actively being infected. Because it's imaging flow cytometry, we can be quite quantitative, and we started doing some time series. And here, what you are looking at is actually the distribution of host cells according to the intensity of the viral messenger RNA. So here at 0 hours post-infection, all of this is done in the lab. all the cells are below this dotted line, meaning that none of them are expressing viral messenger RNA. 4 hours post-infection, you can already see a subpopulation of cells that are extremely bright for this viral messenger RNA, meaning that those cells are actually actively undergoing infection, and we could trace this through time from 0 to 56 hours post-infection. And by getting access to those dynamics of percentage of infected cells, we actually uncovered some quite new aspects in the giant virus's life cycle, namely that we realized that there was an early phase of chronic viral release, so during which host cells are infected. They are still metabolically active, but they start to spit out viruses in the media without lysis, and that this was followed later during the infection by a massive episode of cell death, but with no viral release. So it was quite different from this lytic cycle that we thought about, where viruses enters the cell, starts to replicate, and then the cell lies by releasing virions. Actually, we realized that there's a different mechanism of viral release early during infection. On top of that, because this is imaging flow cytometry, we were also capable of distinguishing let's say, the state of those cells, whether or not they are single cells or particles. So this is a typical FACS plot coming from the imaging flow cytometer, where every dot is an object. And by looking at the DAPI signal, in this case, DAPI area versus its circularity, you can quite easily disentangle single cells from aggregates. And by doing this through time, we were actually also able to quantify the fraction of biomass that existed as single cell versus aggregate, also starting to decipher a bit the relative abundance of the shunt versus shuttle. Also pointing towards a potential defense mechanism, as we saw that some of those aggregates, especially early during infection, are actually enriched in infected cells that could potentially draw down newborn virions and limit the spread to the free living population. So that was mostly done in the lab, but of course we wanted to see to what extent we could expand this to the natural environment, and decided to run a mesocosm experiment. Our mesocosm took place in Norway in May 2018, and it consisted of 7 huge bags of 11,000 liters. Those bags were filled with natural seawater on the first day of the experiment. They were filled with nitrate and phosphate and followed for 23 days with the hope of triggering an algae bloom that we could then follow through time and maybe test whether or not we could evaluate the impact of viral infection on those complex ecosystems. So we were quite lucky because we actually experienced a massive bloom of coccolithophores algae in all of the 7 bags. This is what you see here, based on flow cytometry as a function of time. All the different lines are the different bags, and we are looking at cell abundance. So all of the bags experienced the coccolithophore bloom, and by using viral qPCR, we were also able to quantify the presence of the EHV virus, the Emiliana-Huxley virus. And what you see here is that we have a gradient also across the different bags. We have bags in which we barely see any viruses, even though the bags crashed. And we have bags in which we see a lot of viruses, and also in this case, the bags crashed. So for us, it was actually quite amazing because we had 7 mini blooms. All of them experienced a coccolithophore bloom. All of them crashed pretty much at the same time, but apparently not for the same reasons, given that some of them had viruses and some of them didn't. So that was kind of a quite nice setup in which we could actually potentially assess or at least characterize the impact of viral-associated demise on complex communities, because we had 7 coccolithophore blooms, but different types of associated demise. So what we did was, of course, to follow the community dynamics. What you are looking at right now is an average across the 7 different bags of protist. In this case, you can clearly see the Emiliana-Huxley relative abundance increasing towards the middle of the bloom and then decaying. But of course, beyond viral qPCR, we of course wanted to know how many cells were actively infected during those blooms. And so we decided to use the SM-FISH approach that I presented previously by also modifying it a bit by adding a large subunit 28S in situ hybridization probe to specifically target Emiliana Huxley. So we could take our mixture of cells, we hybridized and specifically labeled the Coccolito 4 cells using the 28S markers, and then we ran the downstream imaging flow cytometry procedure to actually reach those numbers of percentage of infected cells during a bloom. So here we are looking at time, days, and at the percentage of our host cells that are actively undergoing infection. And it was actually quite interesting because it was the first time we actually got those numbers, didn't really know what to expect. And it became very obvious that levels of infection are actually never really above 25% of the population, even though the whole population crashes. And once this was done, what we really wanted to try was to link what is happening at the single cell level with what is happening more at the, let's say, biogeochemical level, mostly by trying to connect that single cell data with bulk measurements of TEP. So TEP stands for transparent exopolymer particles. These are phytoplankton-produced polysaccharides that actually play a major role in carbon cycling. And we were quite lucky to have the lab of Raphaël Simot quantifying TEP accumulation through time in the 7 different bags. And this is exactly what you see here. And so because this is more of a specialized audience, I did put a few details on how we tried to build that link to see if we could better assess the impact of a tiny microbial interaction with a larger ecosystem process, such as TEP accumulation. So what we started by doing was to build a model. This is real data. This is real TEP accumulation through time. But we decided to try and model TEP accumulation as a function of different phytoplankton abundance, mostly Emiliana Huxley, other naked phytoplankton, picoeukaryotes that were all estimated using flow cytometry and imaging flow cytometry. And then there was a factor for degradation and loss through sinking. So we built that TEP model. Once we had built that model, we moved to the next step, which was that we can actually, thanks to this model, evaluate how much is being produced by all Emiliana Huxley cells together. And we plotted this number against the total number of viruses per bag. So here, this TEP per cell corresponds to the amount of TEP that is produced per Emiliana Huxley cell in the different bags. So these are our 7 different bags. And then what we just did was to plot it against total viral abundance. So what you can see straight away is that there's a trend. We thought that was quite interesting because it kind of suggested that E. Huxley cells in bags that had a lot of viruses potentially produced more TEP. What you can do from this graph is actually take the cells that are down here. These are bags that basically don't have any viruses. And this actually gives you the amount of TEP that is produced by non-infected E. Huxley cells. So we take that number and we put it aside. At the same time, we used the SM-FISH approach to quantify the fraction of infected E. Huxley cells in all of the bags. We did it mostly for, not in all of the bags, excuse me, mostly for bag 2 and bag 4. So we have that estimate of basically the number of E. Huxley cells that are actively being infected. And from combining both of those estimates, we could actually use that data to estimate how much TEP was produced by infected versus non-infected cell. And this is the estimate that we get, that on average, infected cells, infected single cells of Emiliana Huxley could produce 2 to 4 times more TEP than uninfected cells within one single bag. If this was a bit too circumvoluted, it's all detailed in the methods of the paper. But I think that our goal back then was really to try and see if we could link those microbial interactions to biogeochemical processes. We had negative controls. We did a bunch of different tests to make sure that this made sense. But in general, I thought that was quite exciting because I felt it was one of the first time where we could kind of build that link, and for sure modeling has actually played a major role in trying to kind of bridge that scale. So that was actually quite exciting. At the same time, we decided to push a bit further and also conducted targeted and untargeted single-cell transcriptomics in those marine natural samples. One of the studies that was published in Science Advances last year was basically taking natural water from those mesocosm bags. We basically ran to a flow cytometry unit and conducted plate-based targeted single-cell sequencing, specifically gating Emiliana Huxley cells in the flow cytometer. And then we do whole host virus transcriptomics. So the main advantage of conducting whole transcriptomics on single cells is that, of course, you get access to the whole transcriptome. And what we had been able to do was, with support from lab-based data, is that when a single cell expresses a specific viral gene, you can actually quite well say at which stage of viral infection that cell is. So there are temporal marker genes of viral infection that can tell you, okay, this cell is really early during viral infection versus this cell, which is expressing a gene that usually is really a viral gene, which is really usually towards the late stage of infection. So this is what we try to estimate, and this is what you see here. Where this is data coming from day 19. We are looking only at infected cells, and we are looking at the proportion of infected cells that are in early versus late stage of infection. So day 19 is towards the end of the bloom. And as you can see, most of the cells are actually late during infection, which for me made sense because, you know, it's the end of the bloom. Most of the infected cells are probably in late stage of infection. What came to us as a surprise was when you go back in time and that you go more towards day 15, 17, which are actually the beginning of the exponential growth phase of the bloom, and you see that that proportion doesn't change. So even on day 15, which is really at the beginning of the bloom of the algae, you actually have most of the infected cells that are already in late stage of infection. Of course, less cells are infected, but in terms of cell state, it's actually exactly the same proportion. And for us, this pointed towards the idea that most likely you actually have a viral turnover. You have a turnover of viral infection through time, which was something that we hadn't really resolved until now. And that really encouraged us to think about how we have to decouple the fate of individual cells versus the fate of the population. And what I mean by there is that what you have to think about is that very early on during the bloom at the population level, we already have cells that are very late during the infection. And I think we should really think about what is happening at the single cell versus what is happening at the population as kind of two different events. At the same time, in another study that got published a few weeks ago, we conducted untargeted single-cell transcriptomics, so not gating specifically for EHUGs, but actually taking the whole protist community. Back then, the hope was that we could use 10x genomics to discover new pairs of host and viruses, in particular active viruses of host, and it did. So that's quite nice. It means there's good potential in those single-cell transcriptomics approaches to actually discover new pairs of host and viruses, particularly active giant viruses of protists. So the last step was then to reach out to Open Ocean, and here it's very preliminary, nothing is published, but we were lucky in December 2021 to collaborate with the Tara schooner and to use satellite imaging to target the Coccolito fall bloom from space. This was done offshore Patagonia, and the idea was to actually conduct a Lagrangian study where we detected a Coccolito fall bloom from space. We anchored ourselves in the water mass using drifters, and we basically sampled the same water mass for 7 consecutive days to really try and follow the development of this bloom of this same water mass through time. And we collected, of course, all metagenomics, single-cell sequencing, and preliminary data suggest that we did have an Emiliana Huxley bloom. We do have viruses. And of course, the next step is to test whether some of the observations that we had in the mesocosm still hold. So this idea of having a viral turnover in nature, but also to try and see if we can discover other pairs of host and viruses that are infected during this bloom. And yeah, so just to summarize, we tried with Asaf to kind of bring all of this together in a small piece where we think about what are the ways we can study viral infection, marine viral infection across different scales, and most importantly, how we try to connect the dots. But in general, all those quantitative single-cell approaches, be it sequencing, imaging flow cytometry, I think have a lot of potential for the field to discover new pairs of host and viruses, to quantify active viral infection, and not just counting viral particles, but also to evaluate stages of infection. So whether or not cells are early versus late stage. For me, it does raise many questions. How does the fact that we have a turnover of viral infection during a Coccolito 4 bloom impact this shunt versus shuttle concept? This is more of a statement that modeling for us was really the key thing to bridge that gap between actual single cell measurements and actual bulk processes measured in our bank. So that was quite powerful. And then finally, that in general, all those single cell approaches reveal a lot of heterogeneity, specifically related to time and dynamics, and that we should, I don't know how this is going to impact the way we think about more larger scale dynamics in the ocean, but I think this decoupling is really fundamental to better understand dynamics of viral infection in the natural environment. Yeah, so with this, I would like to thank you, and also most importantly, thank Asaf, because most of the work that I presented was done during the postdoc at the Weizmann. Thank you.
Great. Thank you, Flora. That was excellent. Okay, I'm going to open the floor for questions. But I can ask a question to get us started. I'm curious about your high percent of the populations being in late stage infection. And I wondered if the time of day of sampling might have anything to do with that, or if you guys had varied that, or what your thoughts on that are.
Yeah. So could be that the timing of sampling plays a role. We would mostly sample during the morning in the mesocosm study. What is true, though, is that we did the same experiments in the lab. I mean, I didn't. A previous postdoc also did the single-cell transcriptomics in the lab with a much higher temporal resolution. And it was also, he had the same results, that most of the time, the cells very rapidly transition from the early phase of infection to the late. So it's very hard to catch cells in the early phase of infection, and most of the time, most of the infected cells are already late stage. So they transition quite fast into that late stage infection.
Yeah, that's interesting. Thank you. Oh, okay. David has his hand up.
Thank you for that. That was awesome. I was curious, I guess, linking to the first talk a little bit, the data that you have on chronic infections, do you have any thoughts on what environmental parameters might influence the occurrence of chronic infections, and do you have plans to sort of explore that more environmentally?
Yeah. So for sure, the conditions in which we run the lab experiments are not extremely relevant ecologically, because we grow them in F/2 media with a 12-12 day-night cycle, 100 micro-Einstein. So quite extreme in a sense. I'm not planning personally to play with different parameters, but for sure, now that the assay is ready, that was a bit the idea. If I would have had like 10 years ahead, the idea would have been to play with different MOIs, different temperature condition, different nutrient conditions, and then to probe how this affects the dynamics of active viral infection. For sure, I remember doing experiments with very low MOI. So here, all the data that I showed is done at very high multiplicity of infection. So really bombarding the cultures with a lot of viruses to make sure all the host cells are in contact with an infectious particle within 30 minutes. But I also ran a bunch of experiments, not published, but at very low MOI with the same strain of virus and the same host. And it's extremely interesting how it actually, it starts very slow. Of course, you have much less cells that are infected, but, and actually the percentage of cells that get infected is much lower overall. But what is very nice is that because the infection dynamics is slower, the host has much more time to grow. And then even though the percentage of cells infected is lower, the final carrying capacity, I mean, the final viral concentration in the media is actually much, much higher than when you infect at high MOI. So from a viral strategy point of view, it actually makes a lot of sense to, not kill your host straight away, let it grow exponentially. And then even if it's 1% of the population that is infected, if your population is big enough, then you will get much higher viral concentration in the media by the end of the experiment. So yeah, these are things that I would have loved to play with, but no time, unfortunately. Yeah, Jan?
Yes, that was a great talk. I have a question from your mesocosm study. For that, you had peaked in the EHAX abundance for particular bags, but for the viruses, that for that particular bag, the abundance peak was very low. Do you have any thoughts on that?
Yeah, so it depends. What is quite nice is that we have a gradient of scenarios. We have a gradient of scenarios. So we have a specific bag in which the abundance of the Coccolito 4 never reached super high, but the viruses were high. And here what we think happened is that infection started quite early during the exponential growth phase of the bloom. And one of the consequences was that the maximum abundance of the Coccolito 4 bloom just couldn't be reached. So we think that the infection started so early in that bag that here, in this case, the viruses really controlled the magnitude of the bloom. In another bag, we had a much, much higher cell abundance, host abundance, and we also had high viral abundance. But here what we think happened is that viral infection started slightly later. So the host actually had time to divide. And again, a bit what I was saying before, it had more time to divide, less cells were infected, but at the end of the day, we get the same number of virions. So what we figured out is that the timing at which you observe the first infected cell really impacts the magnitude of your bloom. Of course, if viral infection happens very early on, it's going to be very hard for the cells to actually achieve high abundances. So this for sure is something that we observed. I think one big elephant in the room that we haven't really solved is the fact that we cannot really probe for different E. Huxley genotypes. We know that there are different strains of Emiliana Huxley or Gephirocapsa Huxley that are susceptible or sensitive to the virus. And this is not something that we monitor in those bags. The filters are there. This should be done. And I think that this would probably help us really better understand what's happening in those bags if we could monitor those different strains of sensitive versus susceptible cells. Yeah, resistant.
Thank you.
Thank you, everyone. And I want to thank both speakers, Ray and Flora, for their time and these enlightening presentations. And there are a few things that I wanted. So we have the second seminar will be May 20th at 9 a.m. Eastern Time, 8 a.m. Central Time. And also if anyone want to attend the in-person workshop, it's going to be in June Heather put she put the link in the chat. And thank you all for attending.
Thank you. Bye-bye.
Thank you.